Hey guys, so here we are in our first EOC study sessions. We're going to do this in a series of 3 videos. This first one we're going to start from the beginning of the year and work our way through all the way through DNA and RNA. So along with this video, I have a handout for you. I need you to go ahead and pull this up. So grab that so you can go ahead and fill it in. So today we're going to talk about the properties of water, macromolecules, cell theory, cell structure, photosynthesis and cell respiration, mitosis, meiosis, and then DNA replication. And again, all of these things are covered in your study guide packet. This will serve to you for studying basically the first and the whole second quarter of this year to help you with that EOC. So make sure you're filling that out as we go along with this video. So basically, we're going to talk about covalent bonds, and a molecule is a group of atoms held together by covalent bonds, okay? In covalent bonds, the electrons are shared between the atoms. In a water molecule, a covalent bond holds the 2 hydrogen atoms together to that oxygen, and they all share their electrons in this nice little electron cloud, like the picture I have there. Okay? Now, water is considered a polar molecule. Now, because it is a polar molecule, it means that those electrons, yes, they're being shared covalently, but they're not being shared equally. So a polar molecule is a molecule with an unequal distribution of electrons, okay? So what happens is these electrons, they have a negative charge, and as they're floating around in this electron cloud, because that oxygen is bigger, more electrons are found around that oxygen. Because they're negative, that oxygen becomes more negatively charged, and those hydrogens become more positively charged. So this is why water has so many unique properties. It can dissolve a lot of compounds such as sugars and other polar molecules. And we're going to talk about that in just a moment. So when we're talking about water, water is a good solvent, okay? It can dissolve a lot of things. And so a solution is a mixture in which one or more substances are distributed evenly in another substance. The liquid part is the solvent, and the solute are usually the particles that you're trying to dissolve, okay? Okay, so here we are talking about the properties of water. So we have cohesion. That was our first property of water. This is where water molecules are bonded to other water molecules, okay? When they bond together, that's also called surface tension. It kind of forms like a nice little net or layer on top of the water that little insects can run across, okay? It's also why water forms a perfectly round little drop, because water bonds with other water molecules. Then we have adhesion. Adhesion is water molecules bonding to other types of molecules. This is why you see when you pour water into like a test tube or a measuring cup, it dips down in the middle. That's called the meniscus. Okay, because the water's more attracted to the sides of the glass than they are to each other, so it forms that little dip, that meniscus. Also, because of cohesion and adhesion, water is able to move from the roots of plants all the way up to the top of the leaves, kind of like a straw. This is called capillary action, the forces of adhesion and cohesion. Now, water also has a high heat capacity. That means that water resists changes in temperature. So it requires a lot of heat to increase the temperature, and it requires a lot of removal of heat to actually freeze it. Also, water is less dense as a solid, which means that ice will actually float on the top of the water. So here we are on our next section. Okay, this was a thing called macromolecules. Macromolecules are just giant molecules, big molecules. We have 4 categories that make up all living things. We have carbohydrates, lipids, proteins, and nucleic acids. Carbohydrates, they're made out of carbon, hydrogen, oxygen, and subunits called monosaccharides, very simple sugars, okay? Their functions are to be the main source of energy for all living things, okay? Because we need sugar to survive. It gets broken down and turned into ATP in our body, okay? So sugars and starches are examples. Our next category is lipids. It's made of carbon, hydrogen, and some oxygen. We call the subunits of these fatty acids. So we have fats, oils, and waxes being some of the examples. They store energy, protect us, and they surround our cells, forming the cell membranes, okay? Proteins. Proteins are made out of subunits called amino acids. please, please, please don't forget that amino acids and proteins go together. It's very, very, very important. Proteins are super important for our body, which I'll talk about in a second. So they're made out of carbon, hydrogen, oxygen, nitrogen, and some sulfur. They also form structures for our tissues, like our muscles and organs. They help us fight disease. They regulate our cell processes. We have special things called enzymes that speed up chemical reactions. Those are proteins. They also help to transport materials through our cells, so they're super, super duper important. Okay, next one are nucleic acids. Nucleic acids are made out of subunits called nucleotides. And if you remember correctly, nucleotides have 3 parts, right? A sugar, a phosphate, and a nitrogen base. We have carbon, hydrogen, oxygen, nitrogen, and phosphorus. Those are the chemicals that make up nucleic acids. Our examples are DNA and RNA. The NA in RNA and the NA in DNA stand for nucleic acid. And what they do is they store cellular or hereditary genetic information. Okay, so it's DNA and RNA. So an enzyme is a specific protein that speeds up the chemical rate of reaction. And it does this by lowering what we call the activation energy. Any chemical reaction requires the input of energy in order to get going. And so the energy is actually reduced by adding in an enzyme. So basically, an enzyme speeds up our chemical reactions. Now, for enzymes to work properly, they require very, very, very specific conditions. Okay? If they're placed in an environment that is not normal for them, like if they go somewhere where the pH is not the same or the temperature is not the same, then they will become inactive. They will no longer work. Okay, the next thing we covered was a cell theory. And the invention of the light microscope allowed to the development of cell theory. We have 3 parts of the cell theory. Cells are the basic unit of structure and function in all living things. All organisms are composed or made up of 1 or more cells. And then all cells arise or come from pre-existing cells. And those are the 3 parts of the cell theory. Now, when we started talking about cells, we had 2 categories of cells that we covered. We have prokaryotes and eukaryotes. Prokaryotes were very simple looking cells. An example of them are bacteria. If you look at them, remember pro went with no because it did not have a nucleus. We have no membrane-bound organelles, and they have 1 circular loop of DNA. So they have 1 chromosome, and it's just 1 circle. Then we have our eukaryotic cell. We have eukaryotic cells. Okay, examples are plants and animals, protists and fungi. You do because they do have a nucleus and they do have membrane-bound organelles. Their DNA is actually rod-shaped, so it goes straight up and down. Okay. All cells, no matter what kind of cell you are, if you're prokaryote or eukaryote, you have DNA, ribosomes, cell membrane, and cytoplasm. Now we have a lot of different eukaryotic cell structures. Now, a lot of these we've gone over. Really important ones to pick out, the cell membrane. We're actually going to talk just about the cell membrane in a little bit. It allows some things to pass through, keeps other things out. The nucleus, that's where we can find the DNA in eukaryotic cells. Prokaryotes, the DNA is just floating around in the cytoplasm. Ribosomes, this is another really important one. This is where proteins are made. Remember, proteins control basically everything in our body. We have our mitochondria. Mitochondria is the site where cell respiration takes place, where ATP is made. We also, in plant cells, have the chloroplast that captures light energy to do photosynthesis. We have these things called lysosomes, which break apart the nasty stuff in our cell and digest organelles and other things that we no longer need. So those are just some of those parts. What I would like you to do is make sure you fill these in on your study guide, and that way you can go back and review them. Okay, now we're going to talk about the cell membrane. Okay, cell membrane is also sometimes called the plasma membrane. So all cells, no matter what kind of cell you are, you have a cell membrane. Okay, plant cells also have a cell membrane right underneath their cell wall. Okay, so all cells have the cell membrane, and we call it semi-permeable or selectively permeable. And what that just means is it allows some things to pass through, keeps other things out. It's made up of 2 layers. We call it the phospholipid bilayer because it's made up of these phosphates and these lipids. And you can kind of see on that picture here, the phosphates are the round little balls on the top. The lipids are those two little straggly that look like legs, kind of facing the middle. In between some of these layers, we have these bigger like tunnels called protein channels. And the protein channels act as gatekeepers to transport materials in and out of the cell. Small things can kind of flow in between those phospholipids, but bigger things need to use those protein channels to fall through. Now, there's several ways things can move through the cell membrane, and we have several classifications of transport. We have active transport and passive transport. In passive transport, the cell doesn't have to expend any energy. It's just sitting back, letting things move back and forth, okay? Just sitting back, letting it happen. In active transport, the cell actually has to use energy to actually open up and bring something in, or open up to push something out, okay? So, types of transport, we have simple diffusion, okay? These things move from an area of high concentration to an area of low concentration, and they just move in between those phospholipids, back and forth. This is passive transport. Osmosis is a fancy word for the diffusion of water. And again, these move from areas of high concentration to areas of low concentration. Does not require the energy from the cell. It's passive transport. Water just moves back and forth and back and forth. We also have facilitated diffusion. Okay, in facilitated diffusion, this also is a type of passive transport, but bigger molecules actually have to just go in between those protein channels, and the cell, again, doesn't do anything. It's passive transport. It just allows those things to move back and forth. Now, in active transport, we actually actively have to use energy, okay? The cell actually moves things against the high concentration gradient. So it's moving from areas of low concentration, okay, to high concentration. So it's moving things actively. It's using energy. It actually has to push something out or take something in. The next thing we talked about is photosynthesis. Okay, and photosynthesis is the process of changing light energy to chemical energy, and it occurs in plants and some algae. And plants need the light energy from the sun or otherwise, carbon dioxide and water. They take in that energy. That energy gets stored in the form of sugar and glucose. Okay, it takes place in the chloroplast of the cell using chlorophyll, which is a green pigment in the plants, to actually capture that sunlight energy. So over here you can see my chemical equation for photosynthesis. We've got 6 molecules of carbon dioxide along with 6 molecules of water reacting to form glucose and 6 molecules of oxygen. Now that light energy, remember energy is not created or destroyed. Okay, it takes in the energy, it's transformed, and it's held in that glucose molecule. So the energy is there, just waiting to be released. That brings me to my opposite process, cellular respiration. You're going to notice in my chemical equation for this, it's the opposite. We're taking in glucose and oxygen, and then that reacts and forms carbon dioxide and water. And again, we have 1 molecule of glucose, 6 molecules of oxygen reacting to form 6 molecules of carbon dioxide and 6 molecules of water. So basically what happens is we have the breakdown of glucose, okay, to release the energy that was stored there in the form of ATP, adenosine triphosphate. That's the type of energy that all cells use. And this takes place in the mitochondria of the cell. And all living things do cell respiration, even plants. So if we're comparing photosynthesis and cell respiration, they're opposite processes. The energy captured in photosynthesis is used to power cell respiration. Carbon dioxide and water released by cell respiration are used in photosynthesis. Photosynthesis is done by plants. Cell respiration is done by all living things. Okay. The next thing we need to talk about is the cell cycle. Cells go through a period of growth and development before they actually split to form new cells. We call this the cell cycle. So cell cycle is a sequence of growth and division in a cell. So anytime you're getting bigger or making more cells, or maybe you cut yourself and you need to heal, you're going through the cell cycle. And so the majority of a cell's life is spent in a growth period known as interphase. Okay, this is where the cell is growing, getting bigger, making more organelles, and getting ready to divide. Okay, interphase is made up of 3 parts: G1, S, and G2. G1, the cell is just growing. S stands for synthesis because the DNA is being copied. And then G2, the cell grows a little bit more. From there, there goes into a process called mitosis, where the actual cell starts to now split, where the nucleus will divide so that we've got DNA for both sides of the cell. And then following that, we have cytokinesis, where the cytoplasm actually separates the cells into two separate daughter cells. Now, if we have unregulated or uncontrolled cell cycle where the cells are just growing out of control, that can result in cancer. Okay, so mitosis. Now, when our bodies go through the cell cycle, one process they go through is called mitosis. This is how the body makes more cells for growth and repair. Anytime you cut yourself, your cells have to go through this process. So new cells are absolutely identical to their parent cell. So our chromosome number stays the same. So in human body cells, we have 46 chromosomes. When those cells go through mitosis, guess what? Those new daughter cells have 46 chromosomes. They're absolutely identical. Here are the different stages of mitosis. We've got prophase. You can see my descriptions here. We have metaphase, where the chromosomes line down in the middle. We have anaphase, where those chromosomes are starting to move to separate sides. And then the last one, we have telophase, where the centromere split and the sister chromosomes are pulled to opposite pole of the cell and is about to split into two new cells. Okay? Then we have another process, meiosis. Now, I exaggerated a little bit and say meiosis because it's easier for me to keep it in my head that it's separate from mitosis. And I have a little rhyme, so I say meiosis makes me 23 and 23, because meiosis actually is how the body forms gametes, the sperm and the egg cell. And in each human sperm and egg cell, there are 23 chromosomes. You get half your chromosomes from mom, half your chromosomes from dad. So cells going through meiosis start with a diploid number of 46. So human chromosomes 46. Then, when we've gone through these divisions, the gametes, the sperm and the egg, end up with a haploid number of chromosomes, 23 in each, so that when the sperm and the egg join together, we have the total of 46 chromosomes. Here is a little summary. So in meiosis, remember meiosis makes me 23 and 23, we go through two rounds of division. We go through meiosis 1 and then meiosis 2. Just like mitosis, we've got prophase, metaphase, anaphase, and telophase. Okay, if you can remember PMAT, this is the order in which they go: prophase, metaphase, anaphase, telophase. And like I said, they do it just one time in mitosis. Meiosis goes through two rounds of division. So you can see I've detailed some little summaries for each of the stages. So this is prophase 1, metaphase 1, anaphase 1, and telophase 1. Then we're going to go straight into meiosis 2. Okay. Before that, in meiosis 1, we have this thing called crossing over, where the the chromosomes actually wrap around each other and get kind of sticky, and they actually transfer their genes so that they're not absolutely identical in each sperm and egg cell. This is why you don't look absolutely identical to all your brothers and sisters. Okay. From there, we have prophase 2, metaphase 2, anaphase 2, and telophase 2. Okay. When this is all said and done, we end up with 4 non-identical haploid cells, which means they have half the normal amount of chromosomes. So we start with 46 chromosomes, we end up with 23 in each of those daughter cells. And they are absolutely non-identical to each other. And again, this is why you're not absolutely identical to your brothers and sisters. So in sexual reproduction, or the fusion of those haploid cells, sperm and the egg, come together, and this actually increases genetic diversity in organisms because all of the sperm are different. They carry different genes. All those eggs are different. They carry different genes, and when they combine, those genes combine and form different combinations. Okay, next, DNA. So we have deoxyribonucleic acid. DNA is found in the nucleus of eukaryotes and then found floating in the cytoplasm of prokaryotes. Remember, all living things have DNA. It is a nucleic acid made out of subunits called nucleotides, and each nucleotide contains a phosphate, a sugar, and a nitrogen base. The bases in DNA are guanine, cytosine, adenine, and thymine, and we can use the letters G, C, A, and T to represent those. Remember, G always goes with C, and A always goes with T. Okay. The shape of the molecule of DNA is a double helix. You can see that in my picture here. And DNA copies itself through a process called replication. We want to talk about DNA replication. What happens in DNA replication? We have our double helix gets unzipped, okay, forming these things called replication forks. So it actually opens up. DNA polymerase, which is a special enzyme, adds new bases to the strands following the base pairing rules. A's with T's and G's with C's. This happens in multiple steps in the eukaryotic cell until all of the DNA is copied. And the result is 2 DNA molecules that are identical to each other, each containing 1 new strand and 1 original strand. Then we have RNA. RNA stands for ribonucleic acid. It's a nucleic acid just like DNA, except for we have a different sugar in RNA. When we talked about that, the sugar in DNA is called deoxyribose, and the sugar in RNA is called ribose. Now, the nitrogen bases in DNA are also a little bit different. They have the G and the C, guanine and cytosine, and it also has the A, the adenine, but it does not have T. Instead, RNA has a U for uracil. So G's and C's go together and A and U go together. RNA is also a single-stranded molecule, and we have 3 kinds. We have messenger RNA, ribosomal RNA, and transfer RNA. We can abbreviate messenger RNA as mRNA, and it carries the instructions for assembling amino acids. And it actually carries those instructions from the nucleus to the ribosome. The ribosomal RNA, excuse me, or rRNA, forms parts of the ribosome. And this ribosome is actually where the proteins are going to be built. Then we have transfer RNA. Transfer RNA, we can abbreviate tRNA. It carries amino acids to the ribosomes. And they are actually the ones that help to build that protein. RNA synthesis. So how is RNA actually made? RNA is made through a process known as transcription. Okay. So this is a segment of DNA serve as a template to produce a complementary strand. So basically it goes in, it reads the DNA, okay, and it forms this little mRNA message, okay. So it reads it. A's go with now U's, okay. G's go with C's, and so on. So after the RNA is made, it gets sent to the nucleus, or sent from the nucleus to the ribosome, where protein synthesis takes place, or the making of proteins. Okay. So each ribosome reads the RNA message 3 letters at a time, or 3 nucleotides. These 3 are called codons, and each codon codes for a specific amino acid. Remember, amino acids are the building blocks of proteins. So if you look at my example here, I've got a sequence of mRNA. It reads AAC, UGA, CCC, AUA. And again, we read them 3 letters at a time. So my first word is AAC. That forms 1 codon. My second one is UGA. That forms my second codon. Then I have CCC, which forms my 3rd codon. And then I have AUA, which forms my 4th codon. So actually, there's 4 amino acids that is in this message. You can use a thing like a codon chart to actually help you determine which amino acid is supposed to be brought. And if we look at just the first one, we have AAC. We go over here to the first base and we find A. We go up here to the second base to find the other A, and we go over here to the third base to find the U. And then we will know that it was supposed to bring the ASN amino acid to help build our protein. Protein synthesis. So how does the actual protein synthesis happen? So after we have the decoding of the mRNA message, that's called translation. Okay, so the making of the RNA message is transcription and then translation. If you can remember, transcription happens first, translation happens later, because it's the second step. So ribosomes decode the mRNA message and send signals to the tRNA to bring specific amino acids. And each tRNA carries a thing called an anticodon, the complementary base pairs for the codon and an amino acid. And the ribosomes start to assemble these amino acids into long chains to actually make the protein. Just one last little note, the central dogma or the central theory of molecular biology is that DNA makes RNA, which makes proteins. And proteins are what control your entire body. So DNA is basically just the instructions. The RNA helps to build the proteins, and the proteins are what control your body. All right, guys, I'll see you in the next section.